Hello, friend. Welcome to Science for Sleep. Whether you have returned tonight or are discovering this channel for the first time, this is a quiet place to slow down, learn something gentle, and allow the day to fade little by little. Tonight, we are beginning with a plant that appears to break one of the simplest rules of the natural world. It is green, rooted in the ground, and powered by sunlight. Yet it also waits for small animals to step between two open lobes before closing around them. The Venus flytrap is known to science as Dionea muscipula. It grows naturally in only a small region of the United States, along the coastal plain of North Carolina and South Carolina. There, it sits close to wet, sandy ground in open pine savannas, often surrounded by grasses, mosses, and other low plants. A mature flytrap forms a small circle of leaves called a rosette. Each leaf begins as a flat green stalk and ends in a trap made from two hinged lobes. If you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find this calm corner of science, one sleepy listener at a time. Those two lobes are not jaws, even though they may look like them. They are parts of one highly changed leaf. Along the inner surface are thin sensory hairs, usually 3 on each lobe. When a crawling insect or spider bends one hair, the plant registers the movement through changes inside its living cells. A second touch soon afterward can cause the leaf to close in roughly 1/10 of a second. The trap speed is striking because the plant has no muscles, no nerves, and no brain. Its cells can still send electrical signals, but those signals travel through plant tissue rather than through an animal nervous system. For many years, researchers understood that these signals help trigger the trap. They also knew that the curved leaf stored mechanical energy, rather like a bent object waiting to flip into a different shape. The exact physical change that released this energy remained uncertain. Then, on June 11th, 2026, researchers reported a new answer in the journal Science. They found that the walls around cells on the trap's outer surface became roughly 1/3 softer within about 1 second. This quick change allowed the curved leaf to release stored tension and snap into its closed shape. It offered a new explanation for one of the most familiar movements in the plant kingdom. But the closing trap is only the visible part of a much larger story. A Venus flytrap does not capture animals because it has stopped living like a plant. Its green tissues still contain chlorophyll, the pigment that absorbs light. Using that light, the plant combines water from the ground with carbon dioxide from the air. This process, called photosynthesis, produces sugars that store chemical energy. The same basic process supports an oak tree, a blade of grass, and a patch of moss. An insect does not replace the sun. The flytrap may spend days breaking down a captured animal, but the energy that keeps the plant alive still begins with light. What the animal provides is something different. An insect's body contains nitrogen, phosphorus, salts, and other materials that are difficult to obtain from the flytrap's natural soil. These materials can help the plant build proteins, new leaves, flowers, seeds, and even more traps. In that sense, an insect is less like a meal of calories and more like a small package of concentrated fertilizer. This difference changes the central question. The mystery is not why a plant would want more energy. The mystery is why a green plant standing in sunlight, with its roots in wet ground, can still be missing some of the basic materials required to grow. The answer begins beneath the trap, in soil that looks fertile from a distance, but provides surprisingly little of what the plant needs. Sunlight gives a plant the energy to build, repair, and grow. Inside each green leaf, chlorophyll absorbs light and helps drive photosynthesis. During that process, carbon dioxide enters through tiny pores in the leaf, while water travels upward from the roots. The plant uses light energy to join those materials into sugar. That sugar stores chemical energy and also supplies much of the carbon used to build new tissue. A stem, a root, and the wall around every plant cell contain carbon that once entered from the air. This explains how a plant can create much of its own living material while remaining rooted in one place. Several other ingredients must still arrive from the surrounding soil or water. Light contains no nitrogen atoms. It cannot supply phosphorus, potassium, sulfur, magnesium, iron, or the smaller amounts of other elements needed by living cells. These materials already exist somewhere in the environment, and the plant must absorb them in a usable form. Nitrogen has an especially large role. Plants use it to make amino acids, which are joined together to form proteins. Those proteins become enzymes that control chemical reactions throughout the plant. Nitrogen also appears in chlorophyll and in the nucleic acids that store genetic information. A plant with too little nitrogen may produce pale leaves because it cannot maintain enough chlorophyll. Its growth often slows as well, since new tissue requires a steady supply of protein. Phosphorus serves a different set of jobs. It forms part of DNA and the thin membrane surrounding cells. Phosphorus also appears in molecules that carry usable energy from one chemical reaction to another. Without enough of it, a plant may continue living while producing fewer roots, flowers, or seeds. Magnesium occupies the center of each chlorophyll molecule. Iron helps enzymes move electrons during photosynthesis and respiration. Potassium supports the movement of water and helps control the opening and closing of leaf pores. Each element performs a physical job that sunlight alone cannot perform. Carnivorous plants still possess roots, and those roots continue absorbing water and dissolved minerals. In many species, however, the roots are small and grow through soil, where useful nutrients remain extremely scarce. Nitrogen and phosphorus commonly limit growth in these habitats, while potassium can also become difficult to obtain. Captured animals provide an additional route into the plant. Consider a small beetle held inside a trap. Its muscles and other tissues contain protein, which provides a rich source of nitrogen. The nucleic acids inside its cells contain both nitrogen and phosphorus. Its cell membranes include phosphorus-bearing compounds called phospholipids. The fluids inside the body also contain dissolved ions, including sodium and potassium. At first, most of those materials are locked inside structures that the plant cannot absorb. Digestive enzymes solve that problem by cutting large molecules into smaller pieces. Proteins become short chains and amino acids. Phosphorus-bearing compounds release phosphate. Salts dissolve into charged particles that can move through water. Once these materials are small enough, cells along the trap surface can take them in. Scientists sometimes describe carnivorous plants as mixotrophic. The word refers to organisms that combine more than one nutritional pathway. For a carnivorous plant, carbon and most usable energy come through photosynthesis, while part of its mineral supply comes from captured organisms. The insect caught today did not power the motion that captured it. That movement depended on energy the plant had already stored from light. Several days later, nutrients released from the insect may be moved away from the old trap. They can enter a developing leaf, strengthen a root, support a flower stalk, or become part of a seed. A successful capture may also help the plant produce more chlorophyll, allowing later leaves to gather light more effectively. The animal's body becomes raw material for future growth. Seen this way, carnivory fits neatly within ordinary plant biology. The plant continues living through photosynthesis, while using trapped organisms to gather atoms that its roots cannot collect in sufficient amounts. That shortage becomes more surprising when the habitat appears wet, green, and full of vegetation. Yet the ground beneath it releases very little usable nutrition. Minerals reach a plant through the ground, dissolved water, or the bodies of other organisms. For many carnivorous plants, the ground provides far less than its green surface seems to promise. A bog may be covered with mosses, sedges, small shrubs, and scattered flowers. Water can remain close to the surface for much of the year. From a distance, such a place appears fertile. Below the leaves, however, the soil may contain very little nitrogen or phosphorus in forms that roots can use. Waterlogging creates part of the problem. In ordinary soil, small spaces between grains hold both water and air. When those spaces remain filled with water, oxygen becomes scarce. Oxygen moves through water far more slowly than it moves through open air. Roots receive less of it, and many organisms that normally break down dead leaves and release nutrients also work more slowly. Decomposition still happens, though the process can become delayed and incomplete. Plant remains may gather faster than they are fully broken apart. In northern bogs, this slow accumulation can produce deep layers of peat over hundreds or thousands of years. The nutrients remain tied up inside partly decayed material instead of returning quickly to living roots. Acidity makes access even more difficult. Rainwater contains few dissolved minerals, and repeated rainfall can carry useful compounds deeper into the ground or wash them away. Organic acids released by mosses and decaying plants can lower the soil's pH. A low pH means the ground is acidic. Under those conditions, some nutrients become less available to roots even when the required atoms are present nearby. This combination appears in many landscapes where carnivorous plants thrive. Sundews grow across acidic bogs and wet heaths. Butterworts may occupy open fens, where groundwater reaches the surface through mineral soil. Bladderworts live in nutrient-poor ponds, shallow pools, or saturated ground. Tropical pitcher plants can grow on mountain slopes where heavy rain repeatedly leaches minerals from thin soils. Some species inhabit tepuis, the flat-topped sandstone mountains of northern South America. These isolated summits rise above surrounding forests and often carry shallow, highly weathered soils. The Venus flytrap occupies another version of the same basic problem. Its natural habitat, lies within the coastal plain and sandhills of North Carolina and South Carolina. Many populations grow in open longleaf pine savannas or along the edges between wetter ground and slightly drier sand. These boundary areas are called ecotones, places where two habitats meet. The soil can remain moist for long periods, while the surface still receives strong sunlight. Fire helps preserve that balance. Historically, lightning and human burning sent low fires through longleaf pine landscapes every few years. The flames removed shrubs, young trees, and thick plant litter, without usually killing mature longleaf pines. When fire disappears, woody vegetation grows taller and denser. Shade spreads across the ground, and small plants such as the Venus flytrap lose access to the light they need. This is why water alone does not create a useful carnivorous habitat. A trap leaf often performs photosynthesis less efficiently than a broad, ordinary leaf. The plant therefore needs strong light to support the cost of producing sensory hairs, digestive glands, sticky secretions, or hollow chambers. Reliable moisture also matters. A sundew's glue contains a large amount of water. A bladderwort must keep its trap submerged or surrounded by wet soil. A pitcher must maintain fluid inside a leaf that may remain open for weeks or months. Dry air and dry ground can make those structures difficult to maintain. The familiar pattern combines mineral-poor soil with dependable moisture and open sunlight. There are exceptions. The dewy pine, Drosophilum lusitanicum, grows in seasonally dry parts of Portugal, Spain, and northern Morocco. Its long, narrow leaves are covered with sticky glands that capture insects. The plant may grow on dry, stony slopes where summers bring little rain. Deep roots and morning moisture help it survive conditions that would damage many bog species. Its presence shows that standing water is not required for carnivory. Poor nutrition remains the more important thread. Wet bogs, sandy seepages, tropical mountains, and dry Mediterranean soils can all leave roots short of usable minerals for different reasons. Under those conditions, prey offers a valuable addition. That benefit still comes with a price. A leaf shaped into a trap gives up some of the efficiency of an ordinary leaf, and the nutrients gained from prey must repay that loss over the life of the plant. An ordinary leaf has a demanding job long before it becomes part of any trap. Its flat surface gathers light, while tiny pores exchange carbon dioxide, oxygen, and water vapor with the air. Veins carry water and dissolved minerals into the leaf, then move newly made sugars toward roots, stems, and growing tissue. A broad, thin shape helps expose many chlorophyll-filled cells to sunlight without requiring too much material. Carnivorous leaves keep some of these abilities, though their specialized structures change the balance. A sundew covers its leaf with raised glands that produce sticky liquid. A Venus flytrap builds sensory hairs, digestive glands, flexible lobes, and an outer layer able to change stiffness quickly. Pitcher plants roll or fold leaves into deep chambers containing nectar, slippery surfaces, downward-facing hairs, and digestive fluid. Bladderworts create hollow underwater traps with flexible doors and glands that pump water. Every added structure requires cells, proteins, minerals, and stored energy to produce. Specialization can also reduce the amount of useful light gathered by the leaf. A flat leaf presents a wide surface toward the sun. A pitcher curves much of that surface inward, where it receives less direct light. Digestive glands and waxy zones occupy space that could otherwise contain more chlorophyll. Some traps remain highly photosynthetic, while others perform this basic leaf function less efficiently. Scientists describe this balance with a cost-benefit model. The cost includes building the trap, maintaining it, producing nectar or glue, and giving up some photosynthetic performance. The benefit comes when captured nutrients allow the plant to make additional chlorophyll, produce larger leaves, or reproduce more successfully. Carnivory remains useful when the lifetime benefit exceeds the lifetime expense. Broad comparisons have found that carnivorous plants often gain less photosynthesis from each unit of nitrogen or phosphorus invested in a leaf than ordinary plants do. Depending on the species and the measurement used, their photosynthetic nutrient use efficiency may reach only around 20 to 50% of values measured in non-carnivorous plants. This wide range does not describe every trap in the same way. A simple sticky leaf has different costs from a large tropical pitcher. An underwater bladder has different demands from a Venus flytrap sitting in full sun. Even so, the general trade-off appears across many carnivorous groups. The physical construction of a trap may not always be unusually expensive by itself. Many pitchers are made from thin tissue, and sundew leaves can remain small. A larger cost comes from lost opportunity. Nitrogen placed into digestive enzymes cannot simultaneously support more chlorophyll. Leaf area folded into a chamber cannot gather light as effectively as the same area spread flat. The environmental setting therefore decides whether the wager succeeds. Strong sunlight provides enough energy to support a specialized leaf. Reliable water allows delicate tissues and liquid traps to remain active. Poor soil makes each captured package of nitrogen and phosphorus more valuable. Deep shade changes the calculation. A plant receiving little light may gain minerals from prey, while lacking enough energy to use them efficiently. Drought creates another problem, because sticky secretions, fluid-filled pitchers, and tender trap surfaces are difficult to maintain without water. Nutrient-rich soil can remove much of the advantage. Ordinary plants with efficient leaves and strong roots may grow quickly under those conditions, rising above slower carnivorous species and shading them. The return from a capture also extends beyond the trap that made it. After digestion, absorbed nitrogen or phosphorus can leave an aging leaf through the plant's transport system. Those materials may enter a new trap that is still unfolding. Some can move into a lengthening root or a flower stalk. Later, the same atoms may become part of pollen, an ovule, or the tissues inside a seed. A single captured insect may therefore influence growth long after its empty outer shell has blown away. The full value appears across seasons and sometimes across several generations of leaves. This makes a trap a biological investment, shaped by local conditions. Natural selection favors the design when prey-derived minerals improve survival and reproduction enough to repay reduced photosynthesis and continued maintenance. That calculation also helps separate true carnivory from coincidence. Insects may become stuck, drown, or decay on many plants. A genuine nutritional strategy requires those animal remains to enter the plant and produce a measurable return. For animal remains to become useful, several things must happen in the right order. A plant first has to encounter or attract a small organism. The trap must hold it long enough for its body to begin breaking down. Released nutrients then need to cross into living plant tissue. Those materials must finally improve growth, survival, or reproduction in some measurable way. This sequence helps scientists separate true carnivory from simple accident. Many ordinary leaves collect dead insects. A fly may become wedged between leaf hairs, drown in a rain-filled cup, or decay on a rough surface. None of those events proves that the plant receives anything useful. The important step is the transfer of nutrients from the animal into the plant. In a clear example, such as a Venus flytrap, the route is direct. The trap closes, glands release digestive fluid, and the same leaf absorbs part of the dissolved body. Other plants divide the work among several living partners. This makes the boundary of carnivory less sharp than it first appears. A sticky surface may originally evolve as protection from insects that chew leaves or drink sap. Small herbivores become trapped before they can damage the plant. Over many generations, any ability to absorb nutrients from those bodies could provide an added advantage. A different route may begin with a leaf that holds rainwater. Dead insects fall into the water by chance. Bacteria and fungi break them down, releasing dissolved nitrogen and phosphorus. If the leaf can absorb those materials, a simple water cup begins acting like a weak trap, even before the plant produces its own digestive enzymes. These gradual pathways are important because complex carnivory does not need to appear all at once. Capture can become more reliable. Absorption can become stronger. Helpful microbes or animals can become permanent partners. One of the clearest boundary cases grows in South Africa. The genus Roridula includes shrubs with narrow leaves covered in shining drops. At first glance, they resemble large sundews. Both plants catch insects on sticky surfaces, though the substances they produce are chemically different. Sundews use a water-rich glue called mucilage. It remains wet and stretchy, much like a thick, clear gel. Roridula produces a stronger, oily resin. That resin can hold insects during rain because it does not dissolve easily in water. The trapped animals may include flies, wasps, moths, and other insects large enough to provide a useful supply of nutrients. The plant itself does not digest them with the same direct system used by sundews or flytraps. Protein-digesting enzymes are either absent or play a much smaller role on its leaf surface. Instead, specialized bugs from the genus Pimeridea live among the sticky leaves. Their long legs and careful movements allow them to cross the resin-covered plant without becoming trapped. The bugs reach insects that are firmly stuck and feed on them. Afterward, they leave small drops of waste on the leaf. This waste contains nitrogen compounds that are already much easier for the plant to absorb than an intact insect body. The relationship gives each partner something valuable. The plant supplies a hunting surface and a relatively protected home. The bugs receive a steady source of captured prey. Nutrients from those prey eventually enter the plant through the bug's waste. Researchers tested this transfer using nitrogen isotopes. Isotopes are different forms of the same element, with slightly different atomic weights. The ratio between two nitrogen isotopes can leave a chemical signature that helps reveal where an organism obtained its nitrogen. Adult plants of the species Roridula gorgonias showed nitrogen signatures that were much higher than those found in young plants and nearby non-carnivorous vegetation. Young plants had not yet developed the same full relationship with resident bugs. The difference supported the conclusion that mature plants were receiving a large share of their nitrogen from captured insects through their animal partners. The trap, consumer, and digestive step were spread across two species. For this reason, Roridula is often described as indirectly carnivorous. Researchers may still disagree about where the outer edge of the category belongs. Clear cases absorb nutrients from animals and gain a real biological benefit. Borderline examples may capture prey only occasionally or depend almost completely on outside organisms to process it. The distinction remained difficult for early observers, who could easily see an insect stuck to a leaf, but had few ways to prove that part of its body had entered the plant. That proof began to emerge through the patient experiments of Charles Darwin and a small sundew growing in wet ground. Proving that animal nutrients had entered a plant required more than watching an insect disappear. A leaf could hold a dead fly, while rain, bacteria, or simple decay did most of the work. Researchers needed controlled comparisons that separated touch, chemistry, digestion, and absorption. Charles Darwin spent years making those comparisons with insect-eating plants during the 1860s and early 1870s. His book, Insectivorous Plants, appeared in 1875. 16 years after On the Origin of Species was first published in 1859. Much of Darwin's attention rested on a small plant found across damp parts of Europe, Asia, and North America. The round-leaved sundew is known as Drosera rotundifolia. Its leaves usually spread close to the ground in a loose circle. Each round blade is covered with reddish tentacles, and every tentacle ends in a gland carrying a clear, shining drop. These drops make the plant appear wet with dew even during dry weather. The liquid is a sticky secretion called mucilage. A small insect landing on the leaf may touch one gland at first. Its attempts to pull free bring more of its legs or wings against nearby drops. The longer tentacles around the leaf's outer edge can then bend inward toward material held nearer the center. Darwin wanted to know whether this movement was a simple reaction to pressure. He counted the glands on 31 leaves. The average leaf carried 192 glands. The smallest count was 130, while the largest reached 260. This gave him hundreds of separate living structures on which to place carefully chosen materials. He tested tiny pieces of roasted meat and albumin, the protein found in egg white. Other trials used milk, pollen, and several nitrogen-rich substances. For comparison, he placed bits of glass, wood, ash, paper, and other materials onto the glands. Darwin also tested oils, sugar solutions, and drops of plain water. A physical object could cause some response simply by pressing on a sensitive gland. Nutritious material often produced a stronger reaction, or kept the tentacles bent for a much longer period. Rain falling onto a leaf did not usually make the entire trap close around every drop. That difference mattered in a wet habitat. A sundew that reacted fully to each shower would spend much of its time folding around water. The leaf responded more strongly when contact was followed by useful chemistry. Once suitable material remained on a gland, the surrounding tentacles slowly curved inward. This movement could continue over minutes or hours. The sticky secretion also changed. It became acidic, meaning that it contained more hydrogen ions and could support chemical reactions involved in digestion. Protein-rich material gradually softened and dissolved. The process resembled part of animal digestion, though it occurred outside the plant's cells on the surface of a leaf. Darwin examined the glands closely as this happened. Material inside their cells changed after the plant received nitrogen-rich substances. These visible changes help show that the glands were absorbing products released during digestion. The same structures that produce the sticky secretion could take dissolved nutrients back into the leaf. His experiments separated several stages that might otherwise appear to be one event. Contact stimulated the gland. Chemical signals from useful material affected the strength and length of the response. Tentacles repositioned the captured object. Acid and digestive substances broke down parts of it, allowing smaller compounds to enter living tissue. The sundew had become an experimentally demonstrated digestive system. Darwin sometimes used language that compared plant sensitivity with animal processes, though the evidence did not require a brain or conscious choice. Each result could be understood through cells responding to pressure, dissolved chemicals, and signals moving through leaf tissue. The patience of the experiments was as important as the plant's slow movement. A single trapped fly might suggest carnivory. Repeated comparisons with glass, water, protein, and other materials showed how the system actually worked. Darwin could place a Venus flytrap beside a sundew and recognize that both captured animals. Their visible forms could not reveal how closely they were related, or how many times carnivory had appeared elsewhere. Modern family trees built from genetic material would show that plants had reached this unusual way of life along many separate evolutionary paths. Modern genetics has allowed researchers to compare carnivorous plants in a way Darwin could not. Every living organism carries inherited instructions in its genetic material. Closely related species tend to share patterns in those instructions because they inherited them from a common ancestor. Researchers can compare thousands of these patterns and arrange species on a branching family tree. This approach is called phylogenomics, meaning the study of evolutionary relationships through large amounts of genetic information. The resulting tree places carnivorous plants across widely separated branches. Around 800 to 850 species are currently recognized, although the total changes as new species are described and older classifications are revised. They occur on every continent, except Antarctica. Some float beneath fresh water. Others cling to tropical trees or grow on exposed mountain plateaus. Their traps range from tiny underground chambers to pitchers large enough to hold several liters of fluid. These species belong to at least 13 flowering plant families spread across 6 major orders. An order is a broad biological group containing several related families. The large distance between these groups shows that carnivory did not begin once and pass unchanged to every hunting plant alive today. A major study published in 2023 examined genetic material held inside plastids. Plastids are structures within plant cells, including the chloroplasts that carry out photosynthesis. That family tree indicated that full plant carnivory arose independently at least 11 times. Other broad genetic accounts have produced estimates closer to 13 origins. The difference depends partly on which species are sampled and where researchers draw the boundary between separate evolutionary events. Both estimates reveal the same large pattern. Different plant ancestors entered nutrient-poor habitats and developed their own ways of gaining minerals from animals. Scientists call this convergent evolution. Similar environmental pressure can favor similar solutions in separate groups, even when those groups begin with different body forms and different recent ancestors. Pitcher plants provide one of the clearest examples. Species of Nepenthes grow mainly across tropical Asia, Madagascar, northern Australia, and nearby islands. Their leaves often extend into tendrils, with a hanging pitcher formed at the end. North American Sarracenia species build upright tubes that rise directly from the ground. The South American genus Helianthora grows mainly on high table mountains called tepuis. Far away in southwestern Australia, Cephalotus follicularis produces small pitchers close to the soil. All four groups use hollow leaves that guide animals into fluid-filled chambers. Their shared pitcher shape developed independently within separate plant families. The same broad function appeared more than once because gravity, slippery surfaces, and a deep container provide an effective way to retain small animals. Sticky traps also evolved along several branches. Sundews cover moving tentacles with glue. Butterworts spread adhesive glands across broad leaves. The rainbow plants of Australia use narrow leaves crowded with shining droplets. Each lineage reached the flypaper solution through its own evolutionary history. Roughly 98% of known carnivorous species belong to four especially successful families. The bladderwort family contains Utricularia, Genlissia, and the butterworts. The sundew family includes sundews, the Venus flytrap, and the aquatic waterwheel plant. Tropical pitcher plants form another large family, while the American pitcher plant family includes Sarracenia, Helianthora, Phora, and the California cobra lily. Venus flytraps and waterwheel plants share a close family connection. The waterwheel plant, Aldrovanda vesiculosa, floats underwater and carries small snap traps arranged around a slender stem. Its trap resembles a miniature aquatic version of the flytrap, although water resistance requires a different pattern of movement. Suction bladders developed along another branch within the bladderwort family. Their tiny chambers use pressure differences to pull microscopic animals through flexible doors. These mechanical designs did not appear because evolution was moving toward a planned level of complexity. Each structure survived where it improved the plant's chances of leaving descendants. Sticky leaves and falling pits arose repeatedly because they could develop through gradual changes in surfaces and leaf shape. Snap traps and suction bladders emerged within particular lineages whose earlier structures could be modified into fast-moving systems. Repeated origins also reveal how much of the necessary equipment already existed in ordinary plants. Leaves could change shape. Glands could release liquid or absorb dissolved material. Defense proteins could attack biological tissue, while electrical signals already moved through living cells. Flexible walls allowed leaves to bend, swell, and store mechanical tension. Carnivory grew from those familiar plant abilities as evolution changed where they appeared and how they worked together. Those familiar abilities gave evolution a useful starting point. Nearly every recognized carnivorous trap is built from a leaf, part of a leaf, or a structure closely related to one. The original organ already knew how to gather light, move water, release chemicals, sense damage, and transport dissolved material. Small changes in development could gradually redirect those abilities toward capture. Across different lineages, leaves have taken on several main forms. Sundews and butterworts use adhesive traps coated with sticky glands. Pitcher plants shape leaves into deep pitfall chambers. The Venus flytrap and the aquatic water wheel plant produce snapping lobes. Bladderworts build hollow suction traps beneath water or inside saturated soil. Corkscrew plants form narrow passages lined with inward-pointing hairs that guide microscopic organisms toward a digestive chamber. These structures look very different when fully developed. Each one begins as living plant tissue following instructions stored in genes. Genes contain the information cells use to build proteins and control development. The same set of genes can produce different organs when certain instructions are switched on in one tissue and kept quieter in another. A small Australian plant offers a clear living example. Cephalotus follicularis grows naturally in a limited part of southwestern Australia. It can produce flat leaves and pitcher leaves on the same individual. The flat leaves spread outward and perform photosynthesis efficiently. Its pitchers sit close to the ground as small, lidded cups, often only a few centimeters tall. Both leaf types are produced by the same genome, meaning the plant carries one complete set of inherited instructions. Their shapes differ because separate groups of genes become active during development. A genome study published in 2017 compared those patterns. Flat leaves showed stronger activity in genes connected with photosynthesis. Developing pitchers increased activity in genes involved with shape, wax production, secretion, digestion, and the movement of nutrients. One plant could therefore build a broad solar surface during one stage and a compact animal trap during another. Environmental conditions can also influence which form appears more often. Temperature, season, and the age of the plant affect the balance between flat leaves and pitchers. This gives researchers a useful way to compare carnivorous and non-carnivorous leaf programs within a single species. The proteins used for digestion also reveal how traps evolved. Ordinary plants already defend themselves against fungi, bacteria, and insects. Their cells produce chemicals capable of damaging invading organisms or breaking down injured tissue. Carnivorous plants often activate related proteins inside traps. Chitinase is one example. Chitin is a tough material found in fungal cell walls and in the outer skeletons of insects and spiders. A protein that once helped a plant attack a fungus could later help dissolve part of an arthropod body. Acid phosphatases release phosphate from larger molecules. Ribonucleases cut apart RNA, one of the molecules used by living cells to handle genetic information. Proteases divide proteins into smaller chains that can eventually be absorbed. These protein families occur in ordinary plants as well. Carnivorous lineages changed where the proteins were produced, how strongly they were released, and which versions worked best inside a trap. Gene duplication often helped this process. During duplication, an extra copy of a gene appears in the genome. One copy can continue performing the older task. The second copy is free to accumulate changes over many generations. A defense protein may gradually become better suited to acidic digestive fluid. A transporter once used in roots may become active in a trap gland, where it moves nitrogen or phosphorus into the leaf. Comparisons among unrelated carnivorous plants have found similar changes in digestive proteins. The details arose separately, yet natural selection repeatedly worked with the same useful molecular tools. Leaves were already flexible. Glands could already secrete and absorb liquids. Electrical signals already moved through plant tissue after injury or touch. Carnivory brought these abilities together in new places and new combinations. A few unusual species stretch the pattern. The western false asphodel, Triantha occidentalis, carries sticky carnivorous glands along its flowering stem, rather than on a classic trap leaf. Even this exception uses familiar secretory tissue placed in an unusual location. Building a trap solves only part of the nutritional problem. Its glands and surfaces remain empty until a small animal reaches them. Before digestion can begin, the plant must first influence where that animal lands, crawls, or places its feet. A finished trap still occupies only a few centimeters of a much larger landscape. A sundew leaf may be smaller than a person's palm. A Venus flytrap sits close to the soil among grasses, pine needles, and moss. Even a large hanging pitcher represents only one possible landing place in a forest filled with bark, flowers, leaves, and open air. Chance alone can bring prey within reach. Many carnivorous plants also increase the odds by using signals that insects already follow during ordinary feeding. Nectar provides one of the clearest examples. This sugary liquid usually appears in flowers, where it rewards animals that carry pollen. Carnivorous plants can produce nectar on traps as well. In tropical pitcher plants of the genus Nepenthes, nectar glands often occur beneath the lid and around the raised collar at the entrance. That collar is called the peristome. An ant or beetle may reach the outer rim and feed safely for a time. Each drop encourages it to continue along the same surface. The animal gradually approaches an area where its grip becomes less reliable. This works because insects already spend much of their lives searching for sugar. They find it in flowers, ripe fruit, damaged plants, and the sweet waste produced by sap-feeding insects. A nectar-producing trap enters that familiar feeding system. The visitor does not need to recognize the plant as carnivorous. It simply follows a useful resource. Sticky traps use a different visual effect. The gland tips of a sundew hold rounded drops of clear mucilage. In daylight, hundreds of drops can shine like tiny beads of water. Some may resemble nectar closely enough to attract small flies and other insects seeking moisture or sugar. Once an insect tests the surface with a foot, the appearance of harmless liquid gives way to strong adhesion. Color can also influence approach. Some pitchers display deep red patches, pale windows, or sharp patterns around their openings. Many insects see ultraviolet light, a range beyond human vision. A surface that looks plain to a person may carry stronger contrast through an insect's eyes. The importance of color varies among species, locations, and types of prey. A red pitcher does not automatically catch more animals than a green one. Shade, background vegetation, and the sensory abilities of local insects all affect the result. Scent adds another layer. Certain pitcher plants release smells that resemble flowers, fruit, or fermenting material. These airborne chemicals can travel beyond the physical edge of the trap. A visiting insect may detect them with sensory organs on its antennae and change direction before the plant is visible. Moisture can become part of the same signal, especially in warm habitats where animals regularly search for water. Researchers have also measured increased carbon dioxide inside the cavities of some Nepenthes pitchers. Animals release carbon dioxide when they breathe, while rotting fruit and other biological material can produce it as well. Certain insects use this gas while locating hosts, shelter, or food. A pitcher releasing carbon dioxide may therefore gain an added attraction cue. The effect has been observed in particular species and conditions, so it should be understood as one possible tool, rather than a feature shared by every pitcher plant. Trap position determines which animals encounter these signals. A Venus flytrap remains near ground level. Its traps often meet ants, beetles, spiders, and other crawling arthropods moving through low vegetation. A hanging Nepenthes pitcher may be reached by flying insects or animals traveling along branches. Pitchers formed near the forest floor encounter a different community from those hanging several meters above it. The plant's shape and location quietly filter the possible prey before any capture occurs. Near the entrance, attraction begins to merge with physical guidance. An insect may stand on a stable lid while feeding from nectar glands. Its next steps carry it onto the peristome. When that rim becomes wet, a thin film of water can interfere with the tiny pads used for gripping. Farther down, wax crystals may loosen beneath the feet. Other traps use hairs angled toward the interior, making descent easier than climbing back toward the opening. These signals never remove an animal's ability to turn away. They alter probability by placing rewards along a route that becomes less secure. The trap has already begun to function before the first slip, bend, or touch occurs. For an animal that finally reaches a sticky leaf, rapid movement is unnecessary. Every effort to escape can press another leg or wing into the shining surface. The first contact may involve only the tip of one insect foot. A clear drop stretches between the foot and the leaf as the animal tries to pull away. This liquid is mucilage, a water-rich secretion produced by glands at the ends of sundew tentacles. Its texture allows it to form thin elastic threads. Instead of breaking cleanly, a small fly may lift one leg free and place another into a nearby drop. A wing can brush the leaf while the body twists. Within seconds, several points of contact may replace the first one. The insect's own movement spreads the adhesive across more of its surface. Sundews belong to the genus Drosera, a group containing more than 200 recognized species. They range from tiny plants only a few centimeters across to larger forms with long leaves that rise above surrounding grass. Their traps vary in shape, though the same basic system appears again and again. Glandular tentacles hold the sticky secretion above the leaf. Shorter glands near the center often take part in digestion and absorption. Longer tentacles around the edge can bend toward material held farther inward. This movement develops slowly enough to watch without special equipment. Nearby tentacles may begin leaning toward prey within several minutes. A stronger response can continue for an hour or more. Some species also curl part of the leaf blade around the animal. The Cape sundew, Drosera capensis, has long, narrow leaves that can fold gradually around larger insects. The round-leaved sundew studied by Darwin uses a flatter blade surrounded by longer marginal tentacles. Movement depends on changes inside living cells. Water can shift between cells, altering their internal pressure. Growth may also become uneven on opposite sides of a tentacle. When cells on one side lengthen more than those on the other, the structure bends. The exact balance between pressure changes and growth differs among species and parts of the trap. No muscle is required. Signals produced by touch and dissolved chemicals are enough to change the behavior of the tissue. As the prey remains in place, the secretion takes on a second role. The fluid becomes more acidic and receives digestive enzymes. These proteins cut large biological molecules into smaller pieces. Released nutrients dissolve in the moisture covering the leaf and move into gland cells. The same surface that holds the animal gradually becomes a digestive and absorptive organ. Butterworts use a flatter version of this arrangement. Species in the genus Pinguicula often form rosettes of smooth, pale green leaves. Under magnification, the surface carries two main kinds of glands. Stalked glands rise above the leaf and produce sticky droplets that catch small flies, springtails, and other light prey. Flatter glands closer to the surface release digestive substances and absorb dissolved nutrients. Some butterwort leaves curl slightly along their margins after a capture. The motion remains modest compared with a sundew wrapping tentacles inward. Much of the work happens across the broad leaf surface itself. An insect can become trapped, digested, and absorbed without entering a chamber. Exposed glue must also survive rain. A heavy shower can strike the leaf directly, dilute secretions, and wash away small particles. Leaf angle helps water drain instead of pooling. Fresh mucilage can replace material lost from the glands. The chemistry of the secretion allows it to remain adhesive even in habitats where wet weather is common. A similar shine covers the leaves of Roridula, though its resin behaves differently. Sundew mucilage contains a large amount of water and works directly with the plant's digestive system. Roridula produces an oily resin that resists rain and holds larger insects firmly. Resident bugs then process those captures and return nutrients through their waste. Two sticky leaves can therefore appear almost identical while sending captured material through different biological routes. The sundew's method succeeds because time is available. Adhesion begins the capture. Continued movement strengthens it, and slow bending places more digestive tissue against the prey. A Venus flytrap faces a faster problem. Its visitor may cross the entire leaf in a few steps, so the plant must detect those steps and commit to closure before the animal leaves. Speed changes the problem from slow reinforcement to rapid detection. Inside each open Venus flytrap, the two lobes usually carry 3 trigger hairs apiece. These fine hairs rise from the inner surface where a crawling insect is likely to brush them. A slight bend places strain on sensitive cells near the base of the hair. Their outer membranes contain proteins called mechanosensitive ion channels. An ion is an atom or molecule carrying an electrical charge. When the hair bends far enough, some of these channels open and allow charged particles to move across the cell membrane. That movement changes the electrical voltage of the cell. The first change is known as a receptor potential. If it becomes strong enough, it produces an action potential, a brief electrical signal that spreads through the trapped tissue. Animals also use action potentials, though the flytrap has no nerves. Its signal passes from cell to cell through ordinary plant tissue. One brief action potential usually leaves the trap open. A second arriving soon afterward normally causes closure. At moderate temperatures, the useful interval is often described as roughly 20 to 30 seconds. A raindrop or a small piece of falling debris may bend one hair and produce a single signal. The trap avoids spending energy unless further movement follows before the temporary effect fades. This two-signal rule has useful exceptions. A study published in 2020 showed that one carefully controlled bend could generate two action potentials. The hair had to move in a way that created two separate electrical events during the same disturbance. Under natural conditions, a struggling animal can also bend one trigger hair more than once. The important threshold depends on electrical signals rather than a strict count of separate hairs. After the first signal, calcium ions begin changing inside the trapped cells. Calcium carries information through many living systems. Its concentration may rise briefly in one part of a cell, spread into nearby tissue, and then slowly return toward its earlier level. A second stimulus arriving during this temporary state adds to the remaining effect. Once the combined signal crosses the closure threshold, the lobes move. This short-lasting change is sometimes described as plant memory. It involves electrical and chemical conditions that remain for several seconds. No conscious counting is involved. The cells simply respond differently, while traces of the earlier signal are still present. The signal total continues to matter after the trap shuts. Two action potentials normally begin the rapid closing movement. Additional signals from trapped prey activate more expensive processes. The animal touches the hairs again, while crawling against the enclosed lobes. Electrical activity spreads through the leaf, and plant hormones called jasminates begin guiding the digestive response. Jasminates are also used by ordinary plants after tissue damage or attack by herbivores. A caterpillar chewing a leaf can activate related chemical pathways. The Venus flytrap has connected this old defense system to the capture of prey. After more than 3 action potentials, genes involved in digestion begin increasing their activity. The glands prepare to release enzymes. Transport proteins that will later absorb nutrients also become more active. Repeated movement tells the plant that the object inside is still alive and large enough to justify further investment. A 2016 experiment measured this process using crickets placed inside traps. During the first hour, the insects produced an average of 63 action potentials. Some individual recordings exceeded 100 signals across 6 hours. Each movement supplied more information about the capture. The pattern influenced digestive secretion and the later uptake of nutrients, including sodium from the prey. The response therefore develops in stages. A first signal prepares the system for a possible second. Closure follows once the threshold is reached. Continued electrical activity commits the leaf to sealing, digestion, and absorption. This arrangement limits the cost of reacting fully to wind, water, or a small object with little nutritional value. It also gives the plant a practical way to measure prey activity through physical contact. The signals reveal when the leaf should close and how strongly digestion should proceed. The electrical pathway still leaves the fastest movement unexplained. Some part of the curved leaf must release stored mechanical energy quickly enough to turn those signals into a snap. Once the electrical signal spreads through the trap, a physical change must turn that message into movement. Researchers had already mapped the basic shape of the open leaf. Each lobe curves outward while holding mechanical stress inside its tissues. After triggering, both lobes pass through an unstable middle shape and curve inward around the prey. This sudden reversal is called snap buckling. A thin plastic lid makes a similar motion when pressure causes it to flip quickly from one curved form into another. For decades, the missing piece involved the force that pushed the flytrap across this unstable point. One leading explanation focused on water moving within the leaf. Plant cells contain water under pressure, which keeps many soft tissues firm. This internal force is known as turgor pressure. Changes in water pressure can move leaves and flowers, although most hydraulic movements develop slowly. Researchers proposed that the flytrap might rapidly shift water between cells or between the inner and outer layers of each lobe. One side would swell as the other side relaxed, changing the leaf's curvature. The idea appeared reasonable because plants regularly control shape through water. A trap can close in a fraction of a second, placing a strict limit on how quickly any hydraulic system would need to work. A study published in the journal Science on June 11, 2026, tested the moving leaf in unusually fine detail. Its title was Fast Cell Wall Softening Causes Venous Flytrap Closure. High-speed cameras recorded the changing curve of the lobes. A small instrument pressed into the leaf surface to measure stiffness. This method, called indentation, reveals how strongly a material resists being pushed. Researchers also cut selected trap tissues and watched how the released pieces bent. Those cuts exposed mechanical stress that had already been stored inside the open leaf. Three-dimensional scans measured changes across the curved surface. Water transport tests examined whether liquid could move through the tissue quickly enough to explain closure. Computer models then combined the measurements and tested how different physical changes would affect the trap. The results pointed to the outermost layer of cells. This thin covering is called the outer epidermis. Within about 1 second of triggering, cell walls in that layer became roughly 30 to 40% softer. A cell wall forms a firm shell around each plant cell. It helps control the cell's shape while resisting the pressure of water inside. Softening allows that wall to stretch and change form more easily. The measured change happened too quickly for large-scale water movement across the thickness of the leaf to serve as the main driving process. Water and internal pressure still help living cells maintain their form. Rapidly changing wall stiffness provides the initiating movement. Before the trap is touched, its curved tissues are already mechanically loaded. the inner and outer layers pull against each other, while the open lobe holds a stable shape. Electrical activity following stimulation somehow causes the outer epidermal walls to soften. As that layer becomes more flexible, the earlier balance of forces changes. Stored stress begins altering the lobe's curvature. Once the leaf crosses its unstable middle position, elastic energy carries it rapidly into the closed form. This explains how a plant can prepare slowly and move quickly. Cell growth and tissue development load the trap while the leaf is forming. A small material change later releases much of that stored energy at once. The exact molecules responsible for such rapid softening remain uncertain. Plant cell walls contain cellulose fibers held within a changing mixture of other materials. Enzymes or shifts in acidity may loosen connections within that structure, though the full sequence has not yet been identified. The 2026 finding, therefore, resolved the main mechanical problem, while leaving a narrower chemical process available for further study. Engineers are interested in this type of movement because it does not require muscles, gears, or a continuously running motor. A soft robotic gripper might store elastic energy gradually and release it after a material changes stiffness. Adaptive building surfaces could also shift between stable shapes with a small trigger. These possibilities remain areas of research rather than finished everyday technologies. For the flytrap, the result is immediate and practical. A living leaf changes its material properties, releases stored tension, and closes before a small animal can easily escape. Even then, the capture remains incomplete. Freshly closed lobes form a loose cage with open spaces between their long marginal spines. Continued movement inside will determine whether the leaf reopens or seals itself into a digestive chamber. In the first instant after closure, the flytrap has only completed the fastest part of the capture. The long spines along the edge of each lobe cross over one another, like the bars of a loose cage. Open spaces remain between them. A tiny insect may squeeze through these gaps and escape before the trap tightens. Allowing very small prey to leave can save the plant several days of digestive work for only a slight nutritional return. A larger animal has less room to move. Its legs, wings, or body continue pressing against the sensory hairs inside the closed leaf. Each contact can produce another action potential. These electrical signals confirm that the trap contains something active, rather than a piece of bark or wind-blown debris. Laboratory experiments have often used crickets between about 6 and 12 millimeters long. In one study, the crickets had an average mass of 23.8 milligrams. An animal of that size can repeatedly bend the hairs while attempting to escape. The continued signals change the trap's response. Its lobes press together with greater force. The edges gradually form a tighter seal, and the open air spaces inside become a small enclosed chamber. Plant hormones called jasmonates help control this shift. Ordinary plants use related hormones when a caterpillar chews a leaf or tissue becomes wounded. In the Venus flytrap, the same chemical system has been linked to prey capture. Once the trap receives more than 3 action potentials, genes connected with digestion begin becoming more active. The glands lining the inner surface prepare proteins that can break apart animal tissue. These proteins belong to a broad group called hydrolases. A hydrolase uses water during the chemical process of splitting a large molecule into smaller pieces. The digestive mixture contains several kinds because a cricket's body is made from many different materials. Cysteine proteases and aspartic proteases cut long proteins into shorter chains. A serine carboxypeptidase removes smaller pieces from the ends of those chains. Chitinases attack chitin, the tough material found in an insect's outer skeleton. Phosphatases free phosphate from larger molecules. Nucleases break down DNA and RNA, while phospholipases act on fatty compounds in cell membranes. Researchers have also identified glucanase and several peroxidases in the digestive fluid. As these enzymes appear, glands pump hydrogen ions into the chamber. That makes the fluid acidic and creates conditions in which several digestive proteins work more effectively. The sealed trap gradually fills with a thin liquid spread across the prey and the inner surface of the leaf. Digestion takes place outside the plant cells. Proteins, nucleic acids, and membranes are broken into smaller dissolved compounds. Special transport proteins then move amino acids, phosphate, sodium, and other useful materials across the gland cells. From there, the nutrients can enter the plant's transport system and travel beyond the trap. The timing varies with prey size, temperature, and the condition of the leaf. A commonly described digestion period is about 5 to 10 days. Cool weather may slow the process, while unusually large prey can remain inside longer. A damaged or poorly sealed trap may fail to digest efficiently. The insect does not vanish completely. Soft internal tissues provide much of the useful material. More resistant parts of the outer skeleton often remain. When digestion is finished, the lobes slowly separate. The trap may reopen around a dry shell, fragments of legs, or the empty outline of the original prey. Rain or wind can remove these remains. Each trap has a limited working life. It may close successfully only a small number of times before the leaf darkens and is replaced by younger growth. An empty closure uses stored energy without providing minerals, while a successful capture can repay part of the cost through later leaves and flowers. The whole sequence therefore unfolds as a staged commitment. Initial contact produces a temporary cage. Continued movement brings tighter sealing and digestive chemistry. Absorbed nutrients complete the return. The famous snap may take only a fraction of a second, yet an even smaller trap beneath pond water can open, pull in prey, and close again in less than 1/1000th of a second. Beneath still pond water, a bladderwort carries traps so small that many are easy to miss without close inspection. The genus is called Utricularia, and it contains more than 220 recognized species. Some float freely in lakes and shallow pools. Others grow through saturated soil, cling to wet rock, or live as epiphytes on other plants without taking nutrients from their hosts. Above the surface, many bladderworts produce delicate flowers shaped a little like tiny snapdragons. Below the surface, finely divided green growth carries hollow bladders that are usually only a few millimeters wide. Each bladder is a reusable pressure trap. Before any prey arrives, glands inside the chamber begin pumping water outward. As water leaves, the flexible sidewalls bend inward. The pressure inside the bladder falls below the pressure of the surrounding pond. This difference is called negative pressure, meaning the water outside is pushing more strongly than the water remaining inside. Mechanical models of active traps have used pressure differences close to 15 kilopascals. That is enough to store elastic energy in the bowed walls, while the entrance stays sealed. The doorway behaves as a flexible flap, pressed tightly against a raised threshold, rather than a stiff door turning on a hinge. A disturbance near the entrance causes the flap to buckle. That brief change breaks the seal. Water immediately rushes from the higher pressure pond into the lower pressure bladder. A tiny crustacean, protozoan, mosquito larva, or other nearby organism can be carried through the entrance with the flow. High-speed recordings have measured the main suction phase at about half a millisecond. 1 millisecond is 1,000th of a second, so the capture can be completed in less time than many cameras need to record a single ordinary frame. During that rush, water speed may reach roughly 1.5 meters per second. Acceleration has been estimated at close to 600 times the pull of gravity. The prey experiences this force across a distance of only a few millimeters. The event remains nearly invisible to an unaided observer. One moment the bladder is set, with its walls drawn inward. A fraction of a second later, it is rounded and filled with fresh water. The flexible door closes again as soon as the pressure difference collapses. Digestion then begins inside the chamber. Glands release substances that help break down captured organisms, while bacteria and other microbes may also contribute. Dissolved nutrients move into the bladder wall and then into the rest of the plant. The same trap can prepare for another capture. Glands slowly pump the added water back out. Its walls bend inward once more, restoring the pressure difference and reloading the elastic structure. This reset takes far longer than the suction itself, often many minutes. A trap therefore stores energy gradually and releases it almost instantly. Researchers have also observed bladderwort traps firing without an obvious animal touching the entrance. In laboratory experiments, some release spontaneously after roughly 5 to 20 hours. A spontaneous firing may draw in microorganisms too small to see clearly. It may also refresh the water and chemical conditions inside the chamber. The exact benefit can vary among species and conditions. Bladderworts show how a plant can achieve extraordinary speed through a stored pressure difference and a flexible valve. Slow pumping prepares the trap, followed by a sudden release governed by fluid physics. A close relative called Genlicea reaches prey through a very different design. Its underground traps rely on narrow passages that guide microscopic life inward through shape alone. The corkscrew plants of the genus Genlicea solve capture without pressure, snapping tissue or visible movement. Above the ground, they produce small green leaves that gather sunlight. Some species form loose rosettes only a few centimeters across. Their flowers rise on slender stems over wet grasslands, seepages, and shallow pools in tropical Africa and South America. Most of the trapping system remains hidden beneath the surface. Pale structures extend through saturated soil like fine roots. These structures are called rhizophils, meaning root-like leaves. They perform some of the work normally associated with roots, including taking in water and dissolved nutrients. Their anatomy shows that they are modified leaves built for capturing microscopic organisms. Each rhizophyll has the general shape of an upside-down letter Y. The lower portion forms a swollen chamber known as the digestion vesicle. Farther outward, the structure divides into two narrow arms. Each arm twists into a loose spiral, giving the plant its common name, corkscrew plant. Small openings along the arms allow water and microscopic life to enter. Beyond those openings, a narrow passage leads inward toward the central chamber. The walls carry rows of hairs angled in the same general direction. A tiny organism moving toward the chamber can pass between them. Movement back toward the entrance becomes more difficult because the hairs point against that route. This arrangement is especially effective for creatures large enough to contact the barriers, including nematodes and mites. Nematodes are narrow roundworms, many of them less than a millimeter long. The traps may also contain amoebae, ciliates, and other single-celled organisms. Ciliates swim by beating rows of tiny, hair-like structures along the outside of their cells. Bacteria are smaller still, and the hairs inside the trap cannot guide each bacterial cell in the same direct way. Research published in 2024 examined how the trap's microscopic surface influences these smaller swimmers. The work combined physical experiments with computer simulations of active particles. Active matter is a scientific term for collections of objects that move using their own energy. Swimming bacteria are one example. Each bacterium travels, turns, bumps into surfaces, and changes direction in a way that may look random. The inner surface of a Genlissia trap contains ridges and other tiny structures arranged unevenly. When swimming bacteria meet these shapes, their paths become slightly more likely to continue inward. A single encounter produces only a small effect. Many encounters across a large number of cells create a measurable net flow toward the digestive region. Physicists call this process rectification. Irregular movement becomes biased in one direction because the surrounding structure is not symmetrical. The plant does not need to push the organisms forward. Their own swimming provides the motion, while the trap's geometry influences where that motion leads. Once organisms reach the swollen vesicle, digestive glands line the inner surface. These glands release substances that help break down captured material. Other glands absorb soluble nitrogen, phosphorus, and additional nutrients released into the chamber. Chemical conditions inside may also influence movement. As organisms are digested, dissolved compounds spread through the liquid. A gradient forms when the concentration of a chemical is stronger in one place and weaker farther away. Some microorganisms can respond to such gradients, although the effect differs among species. This may help draw certain larger organisms deeper into the system, while many bacteria are guided mainly by contact with the structured walls. The whole trap works continuously. No action potential needs to trigger a sudden response. No flexible door has to be reset after each capture. The rhizophyll remains in place as water and microscopic organisms move through the surrounding soil. Random swimming, narrow passages, and inward-facing barriers slowly concentrate living material near the digestive chamber. The plant appears motionless from above, while its hidden leaves influence thousands of tiny movements below. A similar one-way principle becomes much larger in pitcher plants. There, a folded leaf creates a visible chamber filled with fluid, slippery surfaces, and enough space to support an entire community of organisms. A narrow passage can guide microscopic prey inward, but a pitcher plant enlarges that same basic idea into a chamber that may hold a visible pool of liquid. Pitcher traps have evolved several times in plants that are only distantly related. Tropical species of Nepenthes hang pitchers from the ends of leaf tendrils. North American Sarracenia grow upright tubes from ground-level rosettes. Heliamphora rises from the high table mountains of South America, while the California cobra lily, Darlingtonia californica, forms curved hooded traps beside cold streams and mountain seepages. In southwestern Australia, Cephalotus follicularis produces small pitchers close to the soil. Some bromeliads in South America also hold water and captured organisms inside tightly arranged leaves. These vessels resemble one another because gravity and smooth surfaces work the same way on every continent. Their similar shapes arose along separate evolutionary branches. In Nepenthes, a pitcher usually forms at the end of a leaf. The flat part gathers light, while a narrow tendril extends outward and supports the hanging trap. At the pitcher's opening is a raised collar called the peristome. Its surface may carry nectar glands and rows of microscopic ridges that run toward the interior. An insect feeding around the rim can remain stable while the surface is dry. Rain, mist, or condensed moisture changes the physics beneath its feet. Many insects cling through tiny pads that need close contact with a solid surface. A wet peristome can become coated by a continuous film of water. That film separates the footpads from the leaf, much as water can lift a tire slightly away from a wet road. Researchers describe the effect as aquaplaning. The insect loses grip and slides toward the opening. Below the rim, another surface may continue the descent. Some pitchers carry a waxy zone made from tiny crystals. These particles can break loose beneath an insect or coat the pads used for climbing. Other species rely more heavily on downward-pointing hairs. A leg moving deeper into the trap passes smoothly along the direction of the hairs. Climbing upward pushes against their pointed ends. Overhanging walls can make the return route steeper still. By the time the animal reaches the lower chamber, several small disadvantages have accumulated. The liquid inside also varies among species. Some pitchers contain a watery mixture of rain and plant secretions. Others produce fluid with viscoelastic properties. A viscoelastic liquid behaves partly like a fluid and partly like an elastic material. When disturbed, it can stretch into long threads instead of flowing apart immediately. This matters because many insects escape water by making rapid strokes with their legs. In the fluid of Nepenthes rafflesiana, those movements meet greater resistance. The animal may remain trapped even after rainfall has diluted the liquid substantially. The chamber's shape keeps the prey away from solid surfaces, while the stretching fluid interferes with swimming. Rain can also become part of the capture itself. Nepenthes gracilis has a lid that acts as a small impact surface. Ants and other insects may stand beneath it while feeding from nectar. When a large raindrop strikes the top, the flexible lid moves sharply downward. The sudden motion can throw insects from its lower surface into the pitcher below. This impact-driven mechanism allows a tropical rainstorm to supply the final force. A picture therefore works through a sequence rather than one perfect feature. Nectar brings an animal near the opening. Moisture weakens its grip on the rim. Wax, hairs, or steep walls limit the climb back. Specialized fluid holds the animal once it reaches the chamber. Different species combine these parts in different proportions. Recent comparisons show that similar composite traps can arise independently when evolution joins together traits that already existed in simpler forms. A waxy leaf surface, a nectar gland, or a water-holding cup may each serve another purpose before becoming part of a coordinated trap. The pitcher eventually brings prey into fluid, but capture alone leaves most nutrients locked inside skin, muscle, cell membranes, and outer skeleton. Those structures must be broken into smaller chemical pieces before the living wall of the pitcher can absorb them. Once prey lies inside the trap, the useful minerals remain locked inside proteins, membranes, genetic material, and the hard outer shell. The plant begins by changing the chemistry of the surrounding fluid. Glands in the trap wall release hydrogen ions, which lower the pH. A lower pH means the liquid has become more acidic. In a sealed venous flytrap, the digestive fluid can reach an acidity close to that found in a human stomach. This acidic setting helps loosen the shape of proteins and create suitable conditions for several digestive enzymes. It can also limit the growth of microbes that would compete with the plant for nutrients. The enzymes do not all perform the same task. An insect or spider contains many kinds of biological material, so digestion requires a collection of specialized proteins. Proteases cut other proteins into shorter chains. Further chemical steps can reduce those chains to amino acids, the small building blocks used to make new proteins. Venous flytrap fluid contains several classes of proteases, including cysteine proteases and aspartic proteases. Their names refer to the chemical tools used inside each enzyme. Chitinases work on a different material. Chitin forms much of an arthropod's outer skeleton. It also occurs in fungal cell walls, which helps explain why ordinary plants already possess chitinases as part of their defense systems. Inside a carnivorous trap, related enzymes help weaken the captured animal's outer covering. Phosphatases release phosphate groups from larger molecules. That phosphorus can later become part of DNA, cell membranes, or compounds involved in moving energy through plant cells. Nucleases break down DNA and RNA. Ribonucleases specialize in RNA, a molecule that helps cells use genetic instructions. Lipases act on fats, while phospholipases break apart phospholipids found in cell membranes. Each enzyme opens access to a different part of the prey. A single digestive protein could never release everything the plant needs. The process is controlled rather than constant. In the venous flytrap, repeated movement from captured prey generates action potentials. Those electrical signals activate jasmonate hormones, which ordinary plants also use after damage or attack by herbivores. Jasmonate signaling increases the activity of genes that produce digestive enzymes. The trap therefore releases more chemical effort after continued movement confirms that a worthwhile animal remains inside. Digestion only completes the first half of nutrient gain. A dissolved amino acid still sits outside the plant until it crosses a living cell membrane. Glands along the trap surface contain transport proteins that move useful solutes inward. These transporters can take up ammonium, amino acids, phosphate, sodium, and other dissolved ions. The absorbed material then enters the plant's internal transport system. Nitrogen may later appear in a new enzyme or chlorophyll molecule. Phosphorus can move into a developing flower or seed. Sodium and potassium may support the electrical and water balance of living cells. This separation between digestion and absorption is essential. Enzymes release nutrients from complex tissue. Transport proteins complete the transfer into the plant. Some material remains too resistant to disappear fully. After a flytrap reopens, dry pieces of exoskeleton may still sit between the lobes. The plant has removed much of the softer tissue and dissolved chemistry, while leaving behind a light outer shell. Similar digestive systems appear in unrelated carnivorous plants. The Australian pitcher plant, Cephalotus follicularis, produces enzymes from many of the same broad protein families found in sundews, flytraps, and tropical pitcher plants. Genetic comparisons show that these lineages reached similar chemical solutions independently. Defense proteins became digestive proteins. Root-like nutrient transporters became active in trap glands. Stress hormones were connected to prey detection and enzyme release. The trap resembles a stomach because both systems acidify material and use enzymes to break it down. Every step still belongs to plant biology, carried out by leaf glands, plant hormones, and proteins inherited from older systems of defense and recycling. A fluid-filled leaf soon contains more than dissolved prey. Bacteria, fungi, tiny animals, and changing oxygen levels can turn the chamber into a small living habitat of its own. Inside certain pitcher leaves, digestion becomes the work of an entire community. The purple pitcher plant, Sarracenia purpurea, grows across a broad part of North America, from the southeastern United States into Canada. Its open pitchers collect rainwater. Dead insects fall into this water, while living organisms arrive through the air, on animal bodies, or with splashing droplets. Over time, each leaf develops into a tiny freshwater habitat. Bacteria begin colonizing the remains of drowned ants, flies, and beetles. Their enzymes break complex tissues into smaller compounds. Fungi may join this first stage of decay, especially around pieces of the outer skeleton that remain difficult to digest. Other residents feed on the growing microbial population. Single-celled organisms called protozoans consume bacteria and small particles. Rotifers, which are microscopic animals with rings of moving hairs around their mouths, filter material from the water. Mites crawl through the fluid and across prey remains. Midge larvae tear larger bodies into smaller fragments, exposing more surface area to bacterial enzymes. One especially adapted resident is the pitcher plant mosquito, Wyomia smithii. Adult females place their eggs in the water held by Sarracenia purpurea. The larvae develop safely inside the pitchers, where they feed on bacteria, protozoans, and suspended particles. Unlike the terrestrial insects that drown after falling into the trap, these mosquito larvae are built for aquatic life. They breathe at the water's surface and complete their early development inside the plant. The mosquito, microbes, and other small animals form a food web. A food web shows how energy and material pass among several species. Midge larvae shred prey. Bacteria carry out much of the chemical breakdown. Protozoans and rotifers feed on those bacteria, while mosquito larvae consume several smaller members of the community. Waste, dead cells, and partly digested material continue circulating through the water. This activity releases nitrogen from insect tissue. Proteins and other nitrogen-rich molecules become ammonium and smaller organic compounds that the pitcher wall can absorb. Phosphorus is also freed from cell membranes and genetic material. The plant shapes this habitat by controlling the leaf's form, fluid level, acidity, and available oxygen. Its residents then perform part of the digestive labor. The California pitcher plant, Darlingtonia californica, supports a similar system in the cold seeps and wetlands of Northern California and Oregon. A study published in 2017 followed microbial communities inside Darlingtonia pitchers for a full year at 2 wild populations. Newly opened leaves began with relatively simple bacterial communities. During the first growing season, bacterial biomass rose rapidly. Some pitchers reached about 100 billion bacterial cells in each milliliter of fluid. Community diversity and the number of carbon compounds the bacteria could process reached their highest levels around the middle of succession, roughly 55 days after the leaves opened. Early pitchers developed along fairly similar paths. As months passed, the communities inside individual leaves became more different from one another. Each pitcher received its own mixture of prey, rainfall, microbes, and tiny animals. The resulting food webs gradually developed individual histories. Their digestive performance also changed through the season. Bacterial composition, total biomass, and diversity were linked with faster prey decomposition. Leaves containing communities that broke prey down more efficiently also tended to absorb nitrogen more effectively. Overall digestive performance reached its highest level during late summer. The age of the leaf therefore influenced both the organisms living inside it and the value they provided to the plant. Microbial help extends beyond water-filled pitchers. In 2024, researchers studied an acid-loving fungus called Acrodontium crateriform, living in the sticky mucilage of sundews. The fungus was found across several sundew species and different parts of the world. Experiments with the spoon-leaved sundew, Drosera spatulata, showed that its presence increased prey digestion. Fungal enzymes worked alongside the plant's own chemistry, helping release nutrients from trapped insects. Carnivorous plants differ in how much digestion they perform directly. Venus flytraps produce a powerful mixture of their own enzymes. Purple pitcher plants depend much more heavily on their aquatic residents. Between these examples lie many shared systems where plant glands and microbes work together. A pitcher can therefore serve as a trap, an absorbing leaf, and a small pond inhabited by several generations of life. This creates a delicate division among the animals nearby. Some become prey, some assist digestion, and others must remain unharmed long enough to carry pollen between flowers. The animals around a carnivorous plant do not all serve the same purpose. Some become prey, others live inside the trap and help with digestion. A third group carries pollen between flowers, allowing the plant to produce seeds. That creates a real biological conflict. Many carnivorous plants depend on insects for pollination. A bee, fly, beetle, or moth may visit a flower, brush against pollen, and carry some of it to another plant. If the traps regularly captured those same visitors, the plant could gain nitrogen while losing future seeds. Evolution has reduced this overlap in several ways. The Venus flytrap keeps its traps close to the ground. Its flowers rise above them on a slender stalk that may reach roughly 20 to 30 centimeters. This height places the flowers in a different part of the surrounding vegetation. Flying insects can approach the white blossoms without walking across the low rosette of traps. The arrangement also protects the flowers from being shaded by the plant's own leaves. Sundews often use a similar design. A low circle of sticky leaves may sit against wet soil, while a thin flowering stem rises above them. Butterwort flowers are also carried on narrow stalks, separated from the broad adhesive leaves below. Pitcher plants commonly hold their flowers above or beside the traps, rather than directly over the openings. Distance lowers the chance of conflict, though it does not remove it completely. A pollinator may still land on a trap. A prey species may still visit a flower. The amount of overlap differs among plant species and habitats. Flowering season can create another form of separation. Some traps are most active before or after peak flowering. The insects moving through the habitat may also change as temperatures rise, rain arrives, or different plants begin blooming. A trap and a flower on the same plant can therefore interact with partly different animal communities across the year. Color and scent add finer divisions. Flowers may advertise nectar or pollen using one set of chemical signals. Traps may release different scents or place nectar along a route leading toward danger. Many insects respond to ultraviolet patterns that people cannot see. A flower and a trap may appear similarly colored to human eyes, while looking quite different to a bee or fly. Height also changes which animals arrive. Ground-level Venus flytraps commonly encounter arthropods moving through moss, grass, and fallen pine needles. Field observations have found ants, beetles, spiders, and grasshoppers among their captures. Despite the plant's common name, airborne flies are only part of its prey. The trap's position naturally favors crawling animals. Its flowers occupy open air above the ground, where a different mixture of visitors is more likely. This separation works through probability rather than individual recognition. The plant does not identify a useful insect and decide to spare it. Over many generations, plants that reduced harmful overlap left more descendants. Flower stalks became taller where height improved pollination. Trap placement remained low where crawling prey provided a reliable mineral return. Scents, colors, and timing could also shift when those differences improved reproduction. Successful captures may even support flowering later. Producing a stalk, opening several flowers, making pollen, and developing seeds all require nitrogen and phosphorus. Minerals absorbed from prey can help fund that reproductive effort. Carnivory and pollination, therefore, remain connected through the plant's nutrient budget, even when the insects involved belong to different groups. The best strategy is not simply to capture as many animals as possible. A useful trap gains nutrients while leaving enough pollinators alive to move genes between plants. Some carnivorous species have taken this division of animal roles much farther. Their pitchers provide safe feeding surfaces, daytime shelter, or places where visiting animals leave nutrient-rich waste behind. On the mountains of Borneo, some pitcher plants gain nutrients from animals that leave the trap alive. Nepenthes lowii grows in cool, wet forests at high elevations, where low clouds often move through the trees. In these montane habitats, ants may be less abundant than they are in warmer lowland forests. That makes the usual supply of insect prey less dependable. Young Nepenthes lowii plants produce lower pitchers close to the ground. These early traps have features suited to catching insects, including a narrow opening and slippery inner surfaces. As the plant climbs into surrounding vegetation, it begins producing upper pitchers with a very different shape. The lid rises above the opening like a curved feeding platform. Glands beneath it produce a thick, sugary exudate. An exudate is a substance released onto the surface of a plant. Mountain tree shrews visit these pitchers to lick the sweet material. The animal places its front feet near the raised lid, while its hindquarters rest directly over the pitcher opening. As it feeds, droppings can fall into the chamber below. The pitcher collects this waste in the same place where a more conventional species would collect drowned insects. A study published in 2009 used nitrogen isotopes to measure the importance of this exchange. The chemical signatures suggested that tree shrew droppings supplied between 57 and 100% of the nitrogen found in the leaves of the mature plants studied. This is called foliar nitrogen, meaning nitrogen contained within leaf tissue. The pitcher provides a reliable food reward. The tree shrew supplies concentrated nitrogen without being captured or injured. A different Bornean species has turned its pitcher into a daytime shelter. Nepenthes hemsliana produces long pitchers that provide suitable roosts for Hardwick's woolly bat. The small bats enter headfirst and rest above the digestive fluid. Their droppings fall into the lower chamber, where released nutrients can be absorbed by the plant. Finding one narrow pitcher in a dense rainforest is difficult for an animal that navigates with sound. Bats use echolocation by sending out ultrasonic calls and listening for returning echoes. Ultrasound has a frequency above the range of normal human hearing. Near the opening of Nepenthes hemsliana is a curved surface that reflects these calls strongly. Researchers tested its importance by comparing pitchers with untouched reflectors, enlarged reflectors, and reflectors that had been removed. The bats approach pitchers with enlarged reflecting surfaces in an average of about 92 seconds. Unmodified pitchers took roughly 182 seconds to locate. When the reflector was removed, the average approach time increased to around 409 seconds. The pitcher's shape therefore acts as an acoustic guide inside the cluttered forest. A bat receives a protected place to sleep during the day. The plant receives nitrogen through waste deposited inside the chamber. Ants form another partnership in the giant pitcher plant Nepenthes bicalcarata. This species grows in the peat swamp forests of Borneo and can produce pitchers with a capacity approaching 2 liters. Its tendrils contain swollen, hollow spaces that house colonies of Camponotus schmitzi ants. The ants move freely across the plant and can swim inside the pitcher fluid. They retrieve large prey, break animal remains into smaller pieces, and remove material that might otherwise rot too slowly. Ant waste and discarded fragments add nutrients to the picture. The colony can also defend young plant tissue from some herbivores and competing insects. These relationships developed because both partners gained advantages that improve survival or reproduction over many generations. No animal had to be swallowed for the plant to benefit. The essential result remained the same. Scarce nitrogen reached the pitcher. A leaf that began as a pitfall trap could become a feeding station, a bat roost, or a home for ants. Its architecture now supported communication and shelter alongside nutrient collection. Such precise systems depend on equally precise surroundings. The correct animal must remain nearby. Rainfall must keep the pitcher functioning, while temperature and forest structure preserve the conditions each partner needs. Specialization can produce an elegant exchange, though it also leaves the entire arrangement exposed when the habitat begins to change. A highly specialized plant cannot simply move into any patch of wet soil when its original habitat disappears. Each species depends on a particular combination of light, water, chemistry, disturbance, and living partners. The Venus flytrap shows this clearly. It is grown in homes and greenhouses around the world, yet its natural range remains limited to parts of North Carolina and South Carolina. Wild plants occur mainly in the coastal plain and nearby sandhills, often within open longleaf pine savannas. These habitats remain moist for long periods without becoming deeply flooded. Their sandy, acidic soils provide little usable nitrogen or phosphorus. Strong sunlight reaches the ground because grasses and low plants dominate the open surface. Fire helps maintain that openness. Historically, lightning and human burning carried low flames through longleaf pine landscapes every few years. Mature pines usually survived, while shrubs and young trees were reduced. When fire is excluded for too long, woody vegetation grows taller. Shade spreads across the ground, leaf litter becomes thicker, and flytraps lose the bright conditions needed to support their costly traps. Protecting a patch of wet ground therefore preserves only part of the system. The water level must remain suitable. Regular fire or careful mechanical clearing must prevent the habitat from closing over. Nearby development cannot drain the soil or redirect the movement of water. A population surrounded by unsuitable land may also become isolated from pollinators and from other flytraps. A conservation account published in 2019 identified about 124 known populations in North Carolina and 3 in South Carolina. Those numbers describe separate population sites rather than every individual plant. Some locations held only small groups, while a few large protected areas contained most of the known flytraps. By the time of a federal assessment announced in 2023, approximately 2/3 of known plants occurred within the 3 largest populations. The United States Fish and Wildlife Service reported that 98% of known individuals were in populations considered healthy. That finding reflected years of surveys, prescribed burning, land protection, and habitat restoration. It did not mean that every threat had disappeared. The agency determined in 2023 that the Venus flytrap did not require listing under the Federal Endangered Species Act at that time. Continued management played a major role in that decision. Prescribed fires kept important sites open. Protected lands reduced development pressure. Private restoration also produced encouraging results. In May of 2021, surveyors found nearly 1,000 flytraps on restored private land in South Carolina. The plants had returned where suitable hydrology and open habitat had been rebuilt. Illegal collection remains a separate pressure. Venus flytraps grow slowly, and removing mature plants can damage small wild populations. North Carolina made poaching flytraps a felony in 2014. People purchasing one can reduce pressure on wild plants by choosing specimens produced in nurseries or through tissue culture. Tissue culture grows many genetically identical plants from a small amount of sterile plant material. This method can supply the horticultural trade without removing individuals from natural habitats. Other carnivorous plants face even narrower limits. Some tropical pitcher plants occur on one island or a single mountain range. A species may occupy only a thin band of elevation, where clouds provide regular moisture and temperatures remain cool. Certain plants depend on one soil type, such as nutrient-poor sandstone or ultramafic rock rich in metals, and poor in many common plant nutrients. Changes in rainfall can dry pitchers or alter the water held inside them. Warming may shift cloud layers higher along a mountain slope. Drainage can remove the saturated soil required by sundews, butterworts, and bladderworts. The loss of tree shrews, bats, ants, or microbial partners can weaken nutritional relationships that took thousands of generations to develop. A trap may be mechanically successful, while the larger system around it becomes unsuitable. Its survival depends on the complete setting that made carnivory worthwhile in the first place. The small flytrap from the beginning now carries a much larger history within its two curved lobes. Its shape depends on poor soil and open sunlight, while water keeps the tissues active and fire preserves the low savanna. Prey supplies minerals. Electrical signals control the response. Softened cell walls release stored energy, and enzymes finish the transfer. Every visible snap rests on a network of conditions extending far beyond the leaf itself. Seen together, those connections change the meaning of the trap. A carnivorous plant remains rooted in the same basic life as any other green plant. Its chlorophyll absorbs sunlight. Carbon dioxide enters through tiny pores in the leaves. Water rises from the ground through vessels inside the stem and leaf. Photosynthesis uses those materials to build sugars that store energy and supply carbon for new tissue. The shortage lies elsewhere. Wet, acidic soil often contains little usable nitrogen or phosphorus. Water fills spaces that might otherwise hold air, slowing decomposition and reducing the release of nutrients. Heavy rain can wash dissolved minerals deeper into the ground or carry them away. Roots continue working in these habitats, though the supply they reach may remain too small for steady growth. Carnivory adds another path. An insect body contains nitrogen-rich protein, phosphorus-bearing genetic material, salts, and other useful compounds. Digestion releases those materials in forms that living plant cells can absorb. The captured animal becomes part of a larger nutrient system powered by sunlight. Across the world, evolution has built that system many times. Around 800 to 850 carnivorous plant species are known. Genetic studies suggest that full carnivory arose independently at least 11 times, with some estimates reaching 13 depending on how plant families and borderline cases are counted. Each origin began with structures that plants already possessed. Leaves could change shape during development. Glands could release liquid and absorb dissolved material. Defense proteins could attack fungi or damage tissue. Electrical signals could pass between cells after touch or injury. Flexible walls could bend, stretch, and store mechanical stress. Different lineages combined those abilities into several recurring trap designs. Sundews and butterworts use adhesion. Pitcher plants rely on falling pits lined with surfaces that weaken an animal's grip. Venus flytraps release stored energy through elastic snapping. Bladderworts create negative pressure and pull prey through a flexible door. Corkscrew plants use one-way geometry to guide microscopic organisms inward. Each system works on its own timescale. A sundew tentacle may bend over several minutes and continue moving for hours. A Venus flytrap can close in roughly 1/10 of a second. A bladderwort's main suction phase may last less than 1 millisecond. Inside a pitcher, prey processing may continue for days, while the microbial community within the leaf changes across months and seasons. Speed forms only one part of the story. Electrical signals inside the Venus flytrap depend on charged particles moving across cell membranes. These action potentials carry information through plant tissue without nerves. The timing and number of signals control how much effort the trap invests. 2 signals usually begin closure. Continued movement activates jasmonate hormones and digestive genes. Enzymes break down proteins, chitin, membranes, and genetic material. Transport proteins then move the released nutrients into the leaf. Other carnivorous plants share that work with living partners. Bacteria and fungi help digest prey inside pitchers and sticky leaves. Resident bugs process insects trapped by Roridula. Tree shrews leave droppings inside Nepenthes loei. Woolly bats use certain pitchers as daytime roosts, while specialized ants live within swollen tendrils and help handle captured material. The route varies, though the nutritional result remains consistent. Scarce minerals move into the plant. The opening contradiction now has a clear shape. A hunting plant uses plant tissues, plant hormones, plant proteins, and plant growth patterns at unusually specialized limits. Its traps extend ordinary plant biology into adhesion, pressure, chemistry, and partnership. Even the familiar snap of the Venus flytrap now carries a more complete explanation. Research published in June of 2026 identified rapid softening in the outer epidermal cell walls as the change that releases stored mechanical tension. The walls become roughly 1/3 softer within about 1 second. That shift allows the curved lobes to cross an unstable point and close. The exact molecular process that softens those walls so quickly remains unknown. This uncertainty is narrow and measurable. The larger mechanism is now visible. Low against the wet ground of a Carolina savanna, one open trap rests among grass and pine needles. Sunlight supplies its energy. Poor soil creates the need. Its curved tissues hold stored mechanical force, while 6 trigger hairs rise from the inner surface. 2 small disturbances may be enough to show that a useful source of minerals has entered the leaf.